Welcome everyone. Here today we will discuss in this class about this seed. Here in the dicot embryo, we have seen how the formation, how development has taken place in the zygote of dicotyledonous plant and how dicot embryo has been formed. So here in this previous class, this is how the dicot embryo, in your textbook what it is given is, this is how the mature dicot embryo will look like. So this is the embryonal axis. This part is the embryonal axis and here these two are the cotyledons. This is the embryonal axis, these two are the cotyledons and here we have the plumule just above the cotyledonary node. So here how we have got the structure? Here in the previous videos we have seen how the development has taken place in the dicot embryo. How development has taken place on the dicot embryo. We have seen this periclinal division was there. Okay, in this pro-embryo periclinal division was there. So inner and outer cells were formed. 1, 2, 3, 4, 5, 6, 7, 8. Octet was there. So inner cell and the outer cell. This was hypophysis because of the repeated divisions of the hypophysis. Okay. Now these are the suspensor cells. So this is the hostorial cell. So this was how the globular embryo was there. So now this part has changed into root cortex. Okay, this has changed into root tip and root cap. Okay, this part will change into root tip and root cap. So here it will be like this formation will be there. Okay, so this is how you will see it and here this part will form the hypocotyle. Okay, and this part, this above part will form the plumule and cotyledons. Plumule and cotyledons. So this globular embryo will change into a heart-shaped embryo. It will change into a heart-shaped embryo. So it just look like a heart. Not, it won't be like this totally. It will be just, it will look like a heart-shaped embryo and we have seen from here this is one cotyledon and this is another cotyledon. Two cotyledons was there. So this area has changed into hypocotyle. This area changed into hypocotyle. So now this looks like this, a little like this. Okay, a little like this. So from here root tip or root cap, I am removing the suspensor here. Okay, so root tip and root cap, here the cortex, not just one, just above this here will be the root cortex. Both of them will form the radicle, that is from where the roots will be arising. This region, this is this region, this one. Okay. This region is the hypocotyle. This one, this region is the hypocotyle. Here the root tip and the root cap. Okay. Here is the root cortex. This one is the root cortex. Okay. And here, from here, this tip is the plumule. From here is the plumule, here this one. From here the plumule arises and this part will change into one cotyledon, this part will change into another cotyledon. So these two are the cotyledons. So the pro-embryo has changed into a heart-shaped embryo. Okay, the upper part of the pro-embryo has given rise to the cotyledons and plumule. So this is the hypocotyle, this is the root part, the radical part, it forms the radical part. Okay, so this basically is where it is present in the embryo sac. It is present, this heart shaped embryo is present in the embryo sac. Okay. So now as the embryo sac is very small, what happens is this cotyledons tends to bend. The cotyledons, this is one cotyledon, na? So this cotyledon show bending. Okay, this cotyledon shows bending, so it will also show bending. So this will also show bending. Okay, and this part will be this plumule. Have you seen this now? This whole part, this whole is the embryo, this whole is the embryonal axis. This whole part is the embryonal axis. So this is the embryonal axis. These two are the cotyledons. This is one cotyledon and this is another cotyledon. These two are the cotyledons and this is the plumule. This is the place, this is the plumule. So if you see this mature embryo, it is like this, with cotyledons bends, both the cotyledons bend to one side. Okay. So this is the embryonal axis, these two are the cotyledons. So here if we see this one, if I draw this, if I remove this and draw, so actually this is the embryonal axis, it won't be this big, I am just for the sake of drawing. This is this one, this part. Okay, this is the embryonal axis and from here two cotyledons are there. This is one cotyledon, this is another cotyledon, and here just, so here this is the point from where the cotyledons are arising. I will make it short here. Let us see this one. I will draw a little bigger. Embryonal axis won't be this much bigger. Okay. So from here the cotyledons are arising. Okay. This place from where the cotyledons are attached to embryonal axis, this place is called as cotyledonary node. Cotyledonary node, these two are the cotyledons, these two are the cotyledons, and this part above this cotyledonary node, the part above this cotyledonary node, this part, okay, this part which is just above this cotyledonary node, this part is called as epicotyle, and this part which is below this cotyledonary node, this is the hypocotyle. Okay, and this tip which is towards this micropylar end, this tip forms the radicle, and this tip which is towards the callosal end, this tip will form the plumule. Okay, so this is the embryonal axis which is enclosed in the seed. So if you see the pea seed, so inside this pea seed, two cotyledons, when you open it, two cotyledons will be there, and inside that cotyledon, this embryonal axis will be there, in which embryonal axis is there. So each seed consists of three parts. One is the seed coat, another is the embryonal axis, and the third is the reserve food. Reserve food in the sense cotyledons, which are also called as the seed leaves. Cotyledons or the seed leaves. Okay, these seed leaves or cotyledons, they store the reserve food material in dicots. In monocots, very little reserve food material will be there, but as of now what I wanted you to explain from this, how this is the mature embryo look like. Okay, and from this mature embryo, this is how, if we see this in the seed, you will see it like this one. So two cotyledons, these two cotyledons, then this plumule which is above this cotyledonary node, From here the cotyledon is there. Now just above the cotyledonary node is the plumule. So epicotyle is there and the plumule is there, and here below this cotyledonary node we have this hypocotyle, and this is the radicle. Below hypocotyle, that is the small part which is called as the radicle. Okay. So when you sow the seed inside the soil, so when from this radicle arises the roots. From this radicle arises the roots, and from the plumule arises the shoot. From the plumule arises the shoot. So this is how mature embryo will look like, and if we see in the seed, all this is the embryonal axis with the cotyledons. So this is the embryonal axis, and these two are the cotyledons. So here this is the embryonal axis. Actually, embryonal axis will not be this big, it will be very small. It will be very small. So from here the shoots will be arising, the stem and all, and from here the roots will be arising when you sow the seed inside the soil. So here seed is nothing but a fertilized ovule. I already said you, after fertilization ovule changes into seed. So a seed is nothing but fertilized mature ovule. Seed is nothing but it is a fertilized mature ovule. Okay. So this is how this dicot seed is there. If you see this in this is the dicot embryo, and this is how the dicot seed look like. This is how the dicot seed look like. If we see in the monocots, I have explained you that last time. Here, if this is the, for example, this is the corn, maize you take. Okay. So here in this, now this endosperm is divided into two compartments. Endosperm is, because here in monocots I said you endosperm is there. So endosperm is divided into two compartments. This is the outer compartment and this is the inner compartment. Endosperm is divided into two compartments. This is the inner compartment, and this is the outer compartment. Okay. So now in this outer compartment, here a layer is present, that is proteinaceous layer is there. This is the proteinaceous layer. It is called as aileron layer. It is a proteinaceous layer. Outer endosperm is this is actually the endosperm part. The seed which we are seeing, it is full of endosperm. Okay. This endosperm is of two types. One is this outer one, which is proteinaceous part, which is called as aileron layer, and inside this also endosperm, the starchy endosperm will be present. Starchy endosperm. This is the outer endosperm, which is made up of proteins, which is called as aileron layer, and this is the inner endosperm, which is starchy in nature. It is starchy, it is proteinaceous. Okay. Endosperm is divided into two compartments. Outer compartment consists of a proteinaceous layer, that is the aileron layer, and inside the starchy endosperm is there. So two types of endosperms, starchy endosperm, proteinaceous endosperm. So the proteinaceous layer of this proteinaceous endosperm is the aileron layer and the starchy endosperm. And just in the little corner of this of the seed, here in this we find this is the embryonal axis. Okay. This is the cotyledon. This is the embryonal axis. So this is how you get the seed. So there is division. There is division at just at this one end, the basal end, you find this embryo, and here is the endosperm, and here is the endosperm. I said you, in few seeds endosperm will be totally utilized. In this type of seeds, in the pea seed, bean seed and all, pulses and all, those are the two seeds. Chana you see, open it, you get two cotyledons. Two cotyledons will be there. So there you won't find any endosperm, because endosperm has been totally utilized by this embryo. But here you find this in monocot, such as in grasses and in this maize. Example, in maize you find this endosperm will be there in the seeds. So in the seed, this is the seed which we will be eating in the maize. This is the seed. Okay, the seed consists of endosperm. Here endosperm is not there, only just the seed. Embryo is there. Cotyledons is there, embryo. Cotyledon is storing the reserve food material here. So here in this also, in the dicot seed also, this is the embryo. In monocot seed also, this is the embryo. But for this embryo, cotyledons are storing the reserve food material. For this embryo, cotyledons are storing the reserve food material, whereas in monocots, cotyledon is not storing any reserve food material. Cotyledon is not storing any reserve food material. It is having endosperm. So when this embryo needs the nutrition, so this endosperm is digested and it will go to the cotyledon, and from the cotyledon it will go to the embryo. Okay, so here in this also, in this like this plumule is there, so this part is the plumule, because this is the embryonal axis in dicots and this is the embryonal axis in the monocot. So this will be a plumule, this will be a radicle, this is the hypocotyle, this is the epicotyle, and this is the cotyledonary node. This is the cotyledonary node and this is the cotyledon. Like these two are the cotyledons. In monocots we have only one cotyledon. In monocots I have said you, the cotyledon is called as scutellum. The cotyledon is called as the scutellum. So this is the scutellum. Okay, and here in this plumule it is covered by a sheath. This is coleoptile. And this is also covered by sheath. This is coleorhiza. I have explained you all in this previous videos itself, but how this embryonal axis will be seen inside the seed, I have shown you this one. Okay, how this embryonal, this is the embryonal axis and these two are the cotyledons, how they are placed. Okay, so if we open it, this cotyledons, if we separate it, these two are the separated cotyledons and this is the embryonal axis, because here in this cotyledons are bent to one side only. Okay, but if I like this, if I keep like this, one cotyledon at one side, another cotyledon at another side, so in between two cotyledons we find this embryonal axis. So this is how this dicot seed is there. This is how we find the monocot seed. So in the next video we will discuss the different parts of the seed. From outside what do we see, from inside, so we have seen this one. What are the things which are there inside the seed, we have seen this, and from the outside what are the structures which we see, we will see in the next video.